Meiosis. Alright, the introduction for meiosis is that there are two parts to meiosis, meiosis I and meiosis II. There are eight total phases combined in meiosis as opposed to 5 in mitosis. They are prophase 1 and 2, anaphase 1 and 2, metaphase 1 and 2, and telophase 1 and 2. However, in meiosis I, there is an interphase, which is also found in mitosis. The result of meiosis I is that it makes 2 haploid gametes, and meiosis II makes 4 haploid gametes. A haploid is half of a diploid, and then that's the result.
It has one set of chromosomes. During prophase 1, chromosomes become visible, they are already duplicated, crossing over occurs in the chiasma, which is the site of crossing over, the nucleolus disappears, the meiotic spindle form in the cytoplasm, and the nuclear envelope disappears. Prophase 1 is typically the longest stage in meiosis 1, and it consumes 90% of the time for the 2 divisions. The duplicated homologous chromosome pairs cross over in the chiasma, and they form a tetrad in the nucleolus. But at the end, the nucleolus disappears. Rahul is a chromosome and Rohit is a chromosome. As they stay next to each other, they form a tetrad. Now they cross over as their genes mix, and the area where their genes mix is called a chiasma. Their duplicated chromosome, the nucleus breaks down, and they prepare for the rest of meiosis 1. During metaphase 1, the pairs of chromosomes become arranged on the metaphase plate and are attached to the now fully formed meiotic spindle fibers. They are connected to the centrioles, which are at opposite poles of the cell. The homologous chromosomes line up in the middle. Spindle fibers from one pole of the cell attach to one chromosome of each pair, and spindle fibers from one side of the pole attached to the homologous chromosome.
Anaphase 1 begins when the 2 chromosomes of each tetrad separate and start moving toward opposite poles of the cell as a result of the action of the spindle. Notice that in anaphase 1, the sister chromatids remain attached at their centromeres and move together toward the poles. A key difference between the mitosis and meiosis is that sister chromatids remain joined after metaphase in meiosis 1, whereas in mitosis they separate.
Okay, Sal and Van are conjoined at the centromere and they are tetrads, and Rahul and Rohit are tetrads. They separate to opposite sides of the cell. They are still joined at the centromere, and this describes anaphase 1.
The homologous chromosomes pairs each the poles of the cell. Nuclear envelopes form around them, and cytokinesis follows to produce cells. This is what happens in telophase 1.
This is what happens during meiosis 1. Meiosis 2 is very similar to meiosis 1 and mitosis. There is no DNA replication at the start of meiosis 2. Meiosis 2 begins without any further replication of the chromosomes. In prophase 2, the nuclear envelope breaks down and the spindle forms. The centrioles duplicate and the chromosome duplication has occurred, and the nuclear envelope breaks down to continue meiosis 2.
In meiosis 2 and metaphase 2, the chromosomes become arranged on the metaphase plate, much as the chromosomes do in mitosis, and are attached to the now fully formed spindle. Each of the daughter cells completes the formation of the spindle. Single chromosomes align on the metaphase plate, much as the chromosomes do in mitosis. This is contrast in metaphase 1, in which homologous pairs of chromosomes align on the metaphase plate.
There are 2 cells, and in metaphase 2, the 4 tetrads move to the center of the metaphase plate. They line up, and in anaphase 2, they separate.
In meiosis 2, anaphase 2, the centrioles separate and 2 chromatids of each chromosome move to the opposite poles on the spindle. The separated chromatids are now called chromosomes in their own right.
The chromosomes line up on the metaphase plate, and during anaphase 2, the chromosomes separate. After telophase 2, cytokinesis occurs, which forms 4 new haploid cells. During telophase 2, a nuclear envelope forms around each set of chromosomes, and cytokinesis occurs, producing 4 daughter cells, each with a haploid set of chromosomes. This is what happens during telophase 2. Genetic variability is essentially why we do not look exactly like our parents. There are three types: independent assortment, crossing over, and random fertilization. Independent assortment occurs during the metaphase. The expression 2n can help us determine the possible combinations of genes. In humans, there are over 8 million combinations. This picture describes independent assortment. As you can see, the two diploids, alternative 1 and alternative 2, their genes are lined up in very different ways, and in result of that, the 4 haploid cells produced at the end of meiosis II are very different. The next form of genetic variability is crossing over. As you can see, there is one cell, and homologous chromosomes are paired together to form a tetrad. After that, the crossing over occurs in the chiasma. As you can see, the chromosomes are crossing over. After division, each cell has its own set of a chromosome, and as you can see, this green chromosome has genes of the black one, and this black one has genes of the green one. This is the process of crossing over. Random fertilization is simply the combination of tons of different kinds of sperm fertilizing one egg. Therefore, it is called random fertilization. Errors in meiosis. One error that can occur is non-disjunction, and that is when sister chromatids fail to separate, and this is usually diagnosed via karyotype. Non-disjunction during meiosis I. Chromosomes are replicated by DNA synthesis, and meiosis I proceeds. Non-disjunction occurs if spindle fibers from the same pole attach to both centromeres of both homologous chromosomes. This will result in both homologous chromosomes moving to the same pole, thus they are not separated, which results in non-disjunction. While completing meiosis I, the cell divides into two cells. One contains an extra chromosome while the other lacks a chromosome. As you can see, cytokinesis occurs, and there is only one chromosome in one cell and 3 in the other. At the beginning of meiosis II, the pairs of sister chromatids line up in the center. In the top cell, spindle fibers from the same pole attach to both centromeres of a pair of sister chromatids. As you can see, the red spindle and the black spindle both attach to the blue chromosome. This will result in non-disjunction. Instead of going to opposite poles, these sister chromatids are pulled toward the same pole. This will be expressed by the equation n-1 and n+1. The cell that has an extra chromosome will be expressed as n+1, and the one that is lacking will be n-1. As cytokinesis occurs, the top left cell has one less chromosome and the top right has one extra. This rules out in the equation n-1 and n+1. The two cells on the bottom are perfectly as should be, and that's why they are labeled as N. Many non-disjunctions are diagnosed via karyotype. For example, trisomy 13 would be Patau syndrome, trisomy 18 would be Edwards syndrome, and the most famous, trisomy 21, would be Down syndrome. As we can see, meiosis is a very complex process. There is meiosis I, meiosis II, and it also promotes genetic diversity, and this has created a world